Hello, everyone, and welcome to today's live broadcast. Evaluation of MICRONAUT S Anaerobes MIC broth microdilution panels with a large collection of clinical samples. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Bruker Daltonik. For more information about our sponsor, please visit their website at bruker.com. So let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing this presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenter, Miriam Cordovana, Scientist, R&D Methods Development, Microbiology and Diagnostics, Bruker Daltonics. For a complete biography on our presenter, please visit the Biography tab at the top of your screen. Miriam, you may now begin your presentation. Welcome.
Thank you very much for this kind introduction, and I would like to thank the organizers for making this event possible. So, ladies and gentlemen, good afternoon. I am pleased to welcome you to this webinar. My name is Miriam Cordovana, and I come from Italy. I have worked in the Bacteriology section of the University Hospital of Bologna, Policlinico Sant'Orsola-Malpighi, for 13 years, dealing with routine practice and research projects in the field of bacterial antibiotic resistance. Previously, I had worked at the University Hospital Careggi in Florence in clinical microbiology and in the human genetic department. In May, I have joined the R&D department of Bruker Daltonik in Bremen, and today I am glad to present in this webinar a study that I performed when I was in Bologna regarding the evaluation of the MICRONAUT system for susceptibility testing of anaerobes. Which types of infections can be caused by anaerobes? Anaerobic bacteria are a fundamental component of healthy microbiota of skin, gut, genital tract, oral cavity, and in general of all mucous membranes. Infections caused by anaerobes can involve virtually any organ and can have very different clinical presentation and severity. Anaerobic infections can be divided in exogenous infections, endogenous infections, and unusual infections. Exogenous infections are mainly caused by members of the genus Clostridium and are acquired by intake, ingestion, wounds from the outside of bacterial spores, contaminated food, or environment. The most common are Clostridium difficile diarrhea, which poses serious issues in hospital hygiene control, botulism, and tetanus, which can be lethal if not promptly treated. More commonly, anaerobic infections are endogenous, often polymicrobial, caused by the invasion of sterile body sites by anaerobes belonging to normal flora, which can turn into pathogens once out of their habitat. They involve more frequently abdomen, pelvis, skin, and soft tissues, oral cavity, and wounds, and they can be severe and lead to sepsis. More rarely, anaerobes are the causative agents of serious pulmonary infections in cystic fibrosis patients, as well as in patients carrying prosthetic joint plants. They can also cause chronic genital infections and infections following animal bites. Anaerobic infections represent a burden for clinical microbiology diagnostics, and the role of anaerobes as causative agents of severe infections, despite well known, is still nowadays neglected and underestimated. This is partially due to the fact that they are traditionally fastidious to process, handle, and test in clinical microbiology routine, as they are often slow-growing and require dedicated incubation conditions. They pose problems from the moment of sampling to transport to cultivation, resulting in a lab-intensive diagnostic and a long turnaround time. In most laboratories worldwide, antibiotic susceptibility testing of anaerobes is performed only for selected cases, as an example, blood cultures or monomicrobial samples, while the other cases are reported as mixed infections, and the antibiotic treatment is based on empiric choice. Despite the incidence of anaerobic infections remains relatively low, they are still associated with high mortality rates. More, an increasing trend in antibiotic resistance has been observed in the last years, with also an increasing number of multi-drug resistant reports. Therefore, susceptibility pattern of anaerobes is always less predictable, and the empiric treatment has become no longer satisfactory. One side of the burden of anaerobic infections regards the mortality rate. In this study, the authors compared the 28-day mortality rate between patients properly treated and patients not properly treated, and the results are shown in the graphic on the right. As we can see in the graphic on the left, the survival rate resulted higher in those patients who underwent an appropriate antibiotic treatment, while it drops significantly among not properly treated patients. P-value is really highly significant. These findings prove that an empiric antibiotic treatment based on the standard guidelines is no more satisfactory, and it is really important to investigate antibiotic susceptibility of the single anaerobes isolates. Another side of the burden of anaerobic infections regards the antibiotic resistance rates. In this slide, I reported the results of the study performed by Professor Nagy and the Schmidt study group for anaerobes, in which the authors compared the resistance rates reported in 3 different studies performed over a 20 years period of time. This study shows a significant and worrying increase over the years in the resistance rates for all the antibiotic molecules investigated. As an example, widely used first-line antibiotics such as amoxicillin clavulanate and clindamycin rose from 1 to 10% and from 9 to 32% respectively. But even if lower, an increase was observed also for second-line antibiotics, such as imipenem. But after this background, which is the state of the art of routine diagnostics? In the last years, the introduction of MALDI-TOF-MS has greatly revolutionized the diagnostics of anaerobes in terms of identification at species level, enabling to save up to 2-4 days in the reporting time in comparison with a classical biochemical method. But currently, AST remains still a gray zone for different reasons, despite several methods are available, because most of them present important drawbacks that makes them not so amenable for daily routine use. The reference method, which is agar dilution, is not applicable for routine testing, as it is lab intensive. Therefore, it is restricted to a few reference centers. This diffusion, which is largely used for other fastidious bacteria, was shown not to be reliable for anaerobes. The most commonly used method is gradient diffusion test, but it is anyhow expensive and time-consuming. On the other side, molecular methods beyond expensive have very limited applications for anaerobes. Currently, broth microdilution, despite being considered a reference method, is recommended by CLSI only for Bacteroides species, until evidence regarding its reliability with other species will be provided. Nevertheless, 2 commercial systems are available, the MICRONAUT system and the Sensitatus system, which differ mainly for the broth used for the inoculum and the panel of antibiotics included. And now we come to the study that we performed in Bologna. In Bologna, we evaluated the performance of the MICRONAUT S Anaerobes MIC test panel with a collection of clinical isolates. The MICRONAUT panel is a standardized broth microdilution method performed in a 96-well microtitration plate. It contains lyophilized antibiotics, and it is suitable for both gram-positives and gram-negatives. The MICRONAUT panel includes 13 antibiotics with proven activity against anaerobes, covering all the antibiotic classes recommended by EUCAST and CLSI. As an example, penicillins, beta-lactams, plus beta-lactamase inhibitors, combinations, carbapenems, fluoroquinolones, tetracyclines, etc. Each antibiotic is present in a broad range of concentrations. Most of antibiotics have 8 two-fold dilutions, while vancomycin S3 and tigecycline S4, and they enable the determination of MIC values and their interpretation accordingly both to EUCAST or to CLSI breakpoints. To inoculate the panel, a modified Wilkins-Chalgren broth is used, which is pre-reduced before the use to better support the growth also of the most fastidious anaerobic species. Now let's take a look at the study design more in details. Overall, 300 non-duplicate clinical isolates were included, corresponding to 30 genera and 82 species, from the most common to less common ones. The strains were collected prospectively from blood cultures, prosthetic joint infections, abdominal infections, and deep wound infections. To achieve the presence of a higher number of different and less common species, the number of Bacteroides fragilis, the most common anaerobic pathogen, was limited to the first 33 consecutive isolates. The ASD by MICRONAUT was performed in parallel with gradient test method, MIC test strips, using the same bacterial suspensions for the inoculum of both methods. All the procedures in this study were performed on bench and in air environment. The procedure to set up the test is summarized in the pictures from left to right. Briefly, a 0.5 McFarland bacterial suspension in 0.9% saline solution was prepared, and 200 microliters of it were used to inoculate the MICRONAUT broth. 100 microliters of inoculated broth were distributed in each well of the panel with a multi-channel pipette. The panel was then sealed and incubated at 37 degrees in anaerobic environment for 24 to 72 hours. After 24 hours of incubation, the panels were visually inspected, and if a growth was observed in the growth control well, the one circled in red in the image, the MIC values were red. The last picture on the right shows a panel in which a Bacteroides fragilis has grown. As it can be observed, the antibiotics are placed vertically in the panel, with the highest concentrations in the upper part. MIC strips test was performed following manufacturer's instructions, using the same antibiotic molecules, a 1 McFarland suspensions and Brucella blood agar. And here I summarize the results of the study. MIC results were interpreted using EUCAST breakpoint, updated to 2019. Results delivered by the MICRONAUT panels were compared with those of MIC strips, and these latter were taken as reference method, despite it is not a reference method, but it was the lab standard method at the time of the study. Thus, discrepancies between the two methods could actually be interpreted in both directions. The performance of the MICRONAUT panel was evaluated in terms of agreement, essential and categorical, and in terms of error rates, according to ISO recommendations. As you can see reported here in the table on the left, the agreement between the two methods was very good, overall higher than 95%. Also, the error rate was very good, with the majority of very measured error, 21 out of 24, restricted to those antibiotics for which one single breakpoint is defined. Furthermore, most of these discrepancies involved a tapenem, for which the breakpoint overlaps with the epidemiological cutoff. Results were also investigated in terms of incubation time required to observe a visible growth in the panels. As it is reported in the table on the right, nearly all the most clinically relevant isolates, Bacteroides, Clostridium, but also a good number of Prevotella and Fusobacteria, grew in 24 hours. Overall, around 75% of all strains grew after 24 hours, while the more slow-growing species required 48 or 72 hours of incubation. In addition, for the most oxygen-sensitive species, such as Porphyromonas and Dialister, incubation was performed in an environment with a lower residual oxygen content than standard, obtained using a double amount of anaerobiosis generator. For 4 of these isolates, it was possible to observe a growth only after 5 days of incubation. In conclusion, to summarize the results of this study, we have that all the isolates but one Porphyromonas successfully grew in the MICRONAUT panel. No problems were observed for gram-positive cocci, in contrast with the literature, as in some past studies, a failure of growth in the microdilution system was reported for this group of anaerobes. And this is also one of the reasons for which this method is recommended by CLSI at the moment only for Bacteroides. Most of the isolates included delivered a result after 24 hours of incubation, and among them, the vast majority of the most clinically relevant ones. An excellent agreement with the lab standard methods was found, and most of discrepancies were restricted to a tapenem, for which the UCAS breakpoint is surprisingly low and overlaps with the epidemiological cutoff. The MICRONAUT system proved to be a very user-friendly method with a short hands-on time. Therefore, it proved to be suitable for routine testing of anaerobes. Summarizing the study, we can say that the most recent data reported in the literature suggest that there is a critical need for robust and reliable methods for susceptibility testing of anaerobes. Given the high mortality rate of anaerobic infections and given the increasing rate of antibiotic resistance, also among these pathogens, likely as a consequence of the same selective pressure that led to the development of the more famous resistance mechanisms among aerobic bacteria. Different methods have been described up to now and are commercially available, but each one of them shows some important drawbacks that hampers their large-scale implementation in clinical microbiology lab. The findings of the study I have presented here show that the MICRONAUT panel represents a reliable and user-friendly method suitable for routine practice. It fits both with UCAS and CLSI recommendation, and including also second-line antibiotics, it provides a comprehensive result in one single test. It is a universal system which showed to support the growth of all anaerobic species tested, including the most fastidious ones. In addition, the fact that the MICRONAUT Wilkins-Chalgren broth does not contain blood makes the system free from reproducibility and manufacturing issues. This fact, together with a simplified logistic, long shelf life, storage at room temperature, results in an easy of implementation in clinical microbiology routine practice. And the method at the end combines the features of reliability of a reference method with the user-friendliness for operators. A paper was published containing all this data of the study, and if you like, you can find all details and more information. This webinar is part of a collection of webinar series of Bruker Microbiology and Diagnostics in Innovation and Product Launches 2020, helping you achieve your goals. You are welcome to join our next webinars, and you can also watch the previous one on demand. Thank you for your attention, and thank you for attending this webinar.
Thank you, Miriam, for that informative presentation. So we will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Miriam, our first question is, how do you manage the workflow for isolates that are not grown after the 24 hours?
Well, for those isolates, after the first 24 hours of incubation, we anyhow take a look at the growth control well. And if there is growth, as I described in the webinar, we go on with reading the plate. But if there is no growth, we simply incubate the plate again in the pouch or in the jar or wherever, in whichever device we have incubated the plate. So nothing complicated to do. Simply re-incubate and continue incubation until the following day.
Perfect. Thank you so much for that clarification. So moving on to our next question. So Miriam, how do you manage the pre-reduction of the broth?
Okay, the pre-reduction of the broth is, the broth has to be pre-reducted for at least 3 hours. So for a routine practice workflow, you can just pre-reduct them in the morning, as an example. You have to take the broth tubes, unscrew the caps, and incubate the tubes in anaerobic environment in order to let oxygen be removed from the broth in an anaerobic environment. And after that, you just put them into the incubator for the production. And after 3 hours, you take them out, just screw them again, and store them ready to be used, hopefully or normally during the daily routine practice. And if they are not used during the same day, in worst case, can also be used the following day, because production is anyhow very, let's say, it works, and one day does not enable to recover all the oxygen content which was removed. So it's very user-friendly again.
Perfect. Thank you so much. So we do have time for a few more questions, but I do just want to remind our audience that those questions we are unable to answer today and those that do come in during the on-demand period will be addressed by Miriam via the email address that you provided at the time of registration. So Miriam, let's move on to our next question. Are there any limitations regarding species?
In this study, all the species that were included grew in the broth. Of course, with the time of incubation, which depends on the features of the species, which is not so much related to the method itself, but from the species features. Since we compare two methods, in one was agar-based, what did not grow in the MICRONAUT panels did not even grow in the agar. We observed a perfect correspondence in terms of time of growing. So therefore, I found no limitation in terms of species. Only, as I mentioned, for very oxygen-sensitive species such as Porphyromonas and Dialister, I use this double amount of anaerobic generation, because these species are really, really sensitive also to few minutes of exposure to oxygen. So, but I would suggest to try to test and to use them with any species, because we saw no limitations. So, as far as we went on with our study.
Perfect. Thank you so much. So, Miriam, what is your impression on the What is your impression on the performance of Wilkins-Chalgron broth, as it does not contain blood?
Well, in my personal experience, I can say that media containing blood for anaerobes makes routine practice not so amenable for operators, because each time you use a Brucella blood-based medium, you have to take care to the temperature of the medium and to the plate, to humidity of the plate. You have to take care that hemolysis has not occurred. So let's say that this process is much easier to be handled and to be used in a real life high throughput laboratory, I could say. So it is exactly like to use another broth, like Müller-Hinton or brain heart infusion broth and so on.
Perfect. Thank you so much, Miriam. So we do have a couple more questions in the queue. Let's go ahead and take a look and see which one you would like to answer next. Okay, perfect. Let's do this one, Miriam. So, and excuse me if I mispronounce anything. How do gram-positives as C. acnes or F. magna behave with this panel?
We have no problems with none of these species. In most cases, Cutibacterium acnes grew in 24 hours, while Phenagoldia magna, in some cases in 24, in other cases in 48. But we observed no failure of growth for none of these species. So they grew without any problem.
Perfect. And, let's move on to this one. Now, how do you plate out your mixed colonies? Do you do singles? Because I think they are other organisms that are present in the plates or specimen type.
Yes, sure. Many of these samples were polymicrobial samples, especially the ones coming from wounds. And we just performed subcultures of isolated colony in order to have a pure subculture. And from the pure subculture, we performed susceptibility testing. So we were sure, after identification at species level, of course. So we were sure it was a pure culture. And we checked the species identification before culturing, before subculturing, and after subculturing. So immediately before setting up susceptibility testing. So we were sure not to have mixed cultures or misclassifications, misidentifications.
Wonderful. Thank you so much, Miriam. And just to remind our audience again, any questions that we don't have time for today, or those that may need a little bit more research, or those that come in during the on-demand period, they will be addressed via your email address when you register for our webinar today. So moving on to our next question. So Miriam, how important it is to work with agar of sheep blood? Some of our users are growing their bacteria on different media before the AST test.
In my experience, in my former lab, we also used horse blood agar, not only sheep blood agar. But in this study, we used sheep blood agar because we had to do something standardized for the publication. But from my point of view and from my previous experience, there should be no difference between other blood-based agars. At least no differences between sheep and horse blood agar, as far as it is my experience in my years in Bologna. So I would suggest just to try maybe just a couple of samples in comparison with horse and sheep, but I'm quite sure to expect no differences in the performance of the method, because it's anyhow not selective media, and as far as bacteria grow, they are in a good fit, let's say.
Wonderful. Thank you so much. So we may have time for just a couple more questions if any of the audience members want to throw some questions in for Miriam today while we still have her. But in the meantime, we want to thank you again, Miriam. As we're waiting for a couple questions, do you have any final comments for our audience?
Yes, I have. Yeah. Yes.
Yes. If you want to go ahead and let our audience know what those are, and give the audience a second to see if they have any last-minute questions for you.
If there are any questions, I am here to reply. I still have time.
Okay.
Just one.
Well, no worries. To anybody who is listening to this presentation on demand, you'll still be able to submit questions via over to Miriam if you would like. Here's a question. So now, is there any instrumentation needed to run these plates, or could we buy them separately?
Oh no, we just use it no instrument for these panels, and we just use them. And hopefully they should be implemented into the routine in Bologna in the next weeks after the tender. And we just decided to buy the panels separately from any kind of possible instrument. Just to buy panels and broths and to read everything manually.
Perfect. And looks like we got another question that just came in. Now, how is the performance of the MICRONAUT compared to the Vitek anaerobic card?
I don't know, because in my former lab, despite we had the Vitek instrument, we decided not to use the anaerobic cards because we found them not so reliable. Sorry to say, but from our former experience, we decided to use for anaerobes gradient diffusion method, and then hopefully move to microdilution, but not Vitek as method.
Okay. Thank you so much for that, Miriam. All right. Oh, we do want to thank the audience for the questions and for joining us today. It was a great presentation. Thank you so much, Miriam. And just again, as a reminder, questions that we don't have time for, those submitted during the on-demand period, they'll all be addressed via the contact information that you provided at the time of registration. Miriam, here's another one. So, is the MICRONAUT much better than this than the Sensire? I'm sorry, I can't pronounce that word. Excuse me.
I cannot tell which is better than the other because I did not test the sensitizer. But my feeling is that, of course, sensitizer is also a good method because it is a microdilution based. But since the broth that they use is Brucella blood agar, so it contains blood and maybe has to be evaluated to see which is the performance with gram-positive cocci, which are the trickiest part of the bacteria. I think that a thorough evaluation has not been performed yet. So what I can say is that MICRONAUT panels work with any species I tested, but I don't have experience about the sensitizer, with the exception of what I read in the literature.
And moving on to our next question. So, if reading is done manually, how do you maintain a consistency in reading by a different lab technician?
Yes, for a subset of samples, the first 50, I read the panels myself, but I also ask another 1 or 2 persons to read them independently. And then we just, one blind from the other, and we just then compared the results of our reading. And I did this for the first 50 strains. For the other, the most tricky ones, the most difficult, for which the growth was more faint and weak, I continued doing this also until the end of the experiment. I always ask somebody else, beyond me, 1 or 2 persons, to read the most tricky panels, and then we compared. And in very few cases, we observe a difference of 1 dilution step between one reading and the other. And it was never significant in terms of categorical agreement. So it seems to be also really robust and not operator depending.
Perfect. Thank you so much. So we do want to thank Miriam again for her time today and her important research. We'd also like to thank LabRoots and our sponsor, Bruker Daltonik, for underwriting today's educational webcast. So before we go, you can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now, everyone. Please join us next week for part 5 of our webinar series, which is fast typing and classification of Streptococcus pneumoniae and hygiene relevant strains. We look forward to seeing you then. Until next time, everyone, stay safe, stay healthy. Bye-bye, everyone.